Albert and Baron Mons are, I guess I'll say he's from the Netherlands, but he's from California via the Netherlands. Baron is at Leiden, where he's a biosemantic professoriate, which is probably the first in the world. And Tom Reinflesch is from MLM, where he has done semantic medline, which I think I've told you about before. Amy is a long-term colleague and friend from NSF, who, when they finally kicked me out of the CIO position, were smart enough to put her into it. So she and I have already been in cahoots about the possibility of applying these technologies to the NSF proposal database, and so forth. Oh, good. We've got more NSFers show up. Welcome. You're just in time. This is the 3 of the 4 people in the NSF Smart Health Program, NSF emphasis into health research. Misha is on leave from Oregon Health Science, right? And their boss, Howard Wackler, is at this meeting on the West Coast. And Will, and the newest member,
Julia.
Dr. Julia, who is, all right, so these are a lot of people in this room that I'm happy to proselytize semantic technologies to. So you're all here to help me for one thing. So don't fail me. And it's also, this is also an important meeting to get these two guys talking to each other. So we've got cross-semantic, semantic expertise, and semantic expertise over here. Some of the best in the world. So without further ado, we flipped the coin, and Baron won the flip. So I'm going to turn it over to him, unless somebody has a comment or a question before we go.
Please. Thank you very much. Very glad to be here again. I've been here several years ago, more in NSF context. But so let me quickly introduce myself on the right side of this slide. As George already said, I am professor in biosemantics as of next week in Leiden University. It's a new chair, and I'll explain what that is. MBIC is Netherlands Bioinformatics Center, where I'm a scientific director. Albert and I and many others have started the Concept Web Alliance, and one of our things that we have developed in that is nanopublication concepts, which I will talk about today, which is very close to what Tom will talk about in semantic medline. And effectively on the right hand side you see a screenshot of a site we have called nanopub.org. According to Miles Exton of Nature Genetica, it sounded like very small beers, but it is really a sort of environment where you can play around with nanopublication before you decide to really take it seriously. And what I want to do is two things. I want to sketch the real context on why did we develop this all and why do we need it. Then get a little bit into the technology, but only at a very high abstraction level. I won't show you any RDF, which will scare most people away immediately. I don't know whether you will show any of that.
No, I won't.
RDF. But one of the things we discussed also this morning with George is that it has been very difficult to explain the semantic web technology to people that are not deep into it. I think it's too geeky. And maybe I'm the right person to talk here because my first picture here is one of my favorites, which I fight every day in bioinformatics, is that we are supporting the biologists with our difficult formulas. They talk sequences, and as you see here, the center is completely empty and there is a lot of misunderstanding. And I think nanopublication is one way to bridge that enormous divide. And now I'm reconnecting.
Disconnecting.
Whatever. Oops, something goes wrong with the technology.
So people actually watching
It's just recording.
Anyway, okay. So to go on, what is the general data? Big data. Data has far outgrown also institutional handling capacity. So everything we do is super institutional, and you probably read this recent article in The Economist that we are now going to be in the Iota period, and that it is totally impossible for any particular life sciences institute to deal with the data that come to us. And that is why we started something in the Netherlands called the Data Integration and Stewardship Center, and all the enabling technology centers in the Netherlands come together to try and tame the big data and the life sciences, and what I'm talking about today is part of that. And it's really acute because even in 2011, 1.8 zettabytes was added to the data sets we are dealing with. And the same article, which I don't have a slide on, predicts that in 2017 we will have a shortage of 190,000 people that have to deal with data. So we better start, educating some people that know how to deal with it. So the life sciences data have by far passed the Large Hadron Collider. They produce about 15 petabytes per year, and next generation sequencing alone produces 50 petabytes per year today, and that's only the starting of an S-curve. And then I'm always teasing the high energy physicists that their data is, we didn't see anything for 5 years. Oh, there was a neutrino, now we missed it. And that is typically very difficult, very different data from highly information-rich data sets like NGS. So I would argue that the life sciences are leading the pack in terms of big data. They're far ahead of the high-energy physicists. And we deal, other than the high-energy physicists, which are semi-computer freaks by themselves, we're dealing with people, biologists, that run away from the screen if the green button is not where they expected it. So that's quite a challenge, okay? So what is the biggest challenge in life sciences is this. We have an incredible number of different levels: genomics, RNA profiling, proteomics, metabolomics, cells and organs, organisms, and now even ecology and everything. They're all created with very, very different techniques. And this is really creating something we pray every day that things go better. And then we have to do that across many different organisms, So it's a highly multidimensional data in many ways. Different technologies that are not compatible with each other, and then all these complexities. Now, what we are trying to do with our disk and MBIC and so on is try to piece all these pieces of the puzzle together across different omics technologies to make sense of the enormous complexity of biological science. And that is not only to produce big data-based Nature papers with 500 authors, as we now see every day, but the end goal is obviously to support the green, the red, and the white life sciences, as we call them in Europe: plant, breeding, everything, medical, and biotechnology. So we work with a lot of companies, 70 companies, up to DSM and all the pharma companies, to help them to understand these data. And the other scary thing, that's the data as such, but this is a little piece of my personal ridiculogram, my personal network of people I work with. Many of them you probably know, and this is only the Semantic Project. So if I only want to talk to these people once a year, that's a day job, and this is what also modern scientists have to learn to operate in highly international multidisciplinary projects to create these Nature papers with 40 authors that we have all the time. So if we measure anything in the omics age, as we call it, this is what we get. Larry Hunter coined a very nice term for that, the ridiculogram, because it doesn't mean anything to anyone. And we call that now not even ignorance-driven science, but ignorance-driven science. We have no idea what we're looking for. We hope that these data throw a hypothesis at us. That is what we're trying to do now. That's a completely different way of doing science. We call it e-science. And what happens is things like this. We get this pathway after doing any measurement. And if I would look at some nodes in this pathway, and we will hear more about it in Semantic Medline, that are close to each other but probably don't have lines between them because there is no co-occurrence in the literature. These might be the new discoveries. And I want to read about any of these nodes here, which are genes and proteins. The average node has 90 papers about it in Medline. Okay? So there's no way I can read about it. So what we want to do, and if we have time at the end, Albert could show some practical application that this is not fake, this works now. If I say I want to zoom in on this part of the network, which could cost me a year of reading, what I want is I want to know who knows about these three concepts there, genes, proteins, or whatever. And as you can see here for my other lectures, I have a real Skype guy behind him, can change the name anytime. And sometimes you can actually call somebody in the audience. I didn't want to do that now. So really what you want to do is go from reading to consulting. But we still need the papers to believe whether any of these connections, and we'll see more about them later, is true or not. Why would I believe it? Then I want to go back to the literature and read. So we will still need the paper. This is a picture from a big project I'm involved in from the Center for Translational Molecular Medicine, a 16 million euro project, to integrate data. And the reason I put this picture up is that I want to make one thing very clear in life sciences. It is not a goal to make everything interoperable, semantically. Because, for example, if you get clinical data, genomics data, proteomics data, they are captured in mass spec versus NGS for all kinds of machines. They're not even pre-processed. Why would you want to make them talk to each other? Then you pre-process the data until you make it associational, like you associate concepts, like this SNP in the genome is associated with this phenotype. Now you have concepts. At that point, you want to do interoperability. You have an entire work package on that, work package 5 in trade, to say, okay, take all these multi-omics data and turn them into one interoperable system. And that's what the pharma needs, that's what everybody needs. So we do this now in this disk thing, where this is just still context, disk thing where we have data experts that already sit with people that, for example, plan something like the genome of the Netherlands, where we sequence 750 families, father, mother, child, then look at all their connections connected to biobanking and all these things. If you plan something like that, and we did that last year in the Netherlands, and we have Netherlight, which is the fastest light path connection in the world. We boast on that. George was in surf in the Netherlands for that recently. And still, for this little project, which is going to be our daily input in 3 years from now, from sequencing alone, rather than being able to pump the data around via our Netherlight, we had a student driving from one university to the other with 180,000 euros worth of hard disks on her backseat in an old car to transport the data, the rough data of this experiment. So it was ill planned, we had lots of fights, and we need to plan these experiments from day one with the data experts involved, and that's one of the things we are setting up structurally now in Europe. Now my research is much more interesting. This is the famous Sawtooth of the edge of chaos, and we discussed a lot about it this morning. I won't go into any details. Suffice to say that this is basically very much what you will hear later on from Semantic Medline. Each dot in this graph represents two concepts and how close they are together. And the red ones are protein-protein pairs, so each dot represents two proteins, and they have no co-occurrence in Medline, so nobody ever said they were connected. The black ones are connected, and if you focus on this little area there, you see that there's a lot of red dots that really contaminate the black ones, and those are the ones we predict to be associated based on millions and millions and millions of interactions in the computer. And some of these concepts move over time closer, and I have a movie where you see two genes moving closer together over time in the concept space until even people understand that they are associated with the disease. They can get more concepts in common, but also what you can do now is to take all kinds of connections in a big network of concepts and remove redundancy and really increase the prediction of new associations. This is really our research. Forget the details, but this is pure in silico knowledge discovery by computer, and we are only interested in the things that you cannot find in Medline because they have not been ever made explicit before. Then we go to the technical part, and let me first give you a historical overview. We started late 2008, early 2009, something called the Concept Web Alliance, just a think tank of like-minded people. Mark Newsom from the US, Manimus Barloch, Carol Goebel. Most people probably not known to you, but people that are in the field know them very well, like Frank van Harmelen, who was the director of the Large Knowledge Collider and a big semantic web guy. So we got some leading people together, and we said, let's try to find a format that is semantic web compliant, but goes beyond the link to Open Data Cloud, because that is a useless thing in my mind, because of the lack of provenance and context. You just have a bunch of triples, and they connect, and you don't know who did it and where they come from, really. And we said, let's find a way to publish individual associations in the concept web. Whether this is life sciences or not doesn't matter, but I will obviously stick to the life sciences because then I know a little bit what I'm talking about. So early on, we came up with this rough idea of nano-publication in the Escience era. That was a paper that we just presented at the Semantic Web Conference in November 2009. And then Jan and Paul Groff, working with Frank van Harmelen, he's the chair of the W3C group on provenance, published The Anatomy of a Nano-Publication, how it looks in RDF. So if you ever want to look at it in RDF, go to that publication. Then in March 2011, we got a huge project from the pharmaceutical industry called Open Facts, the Open Pharmacological Space, Open Pharmacological Concept Triple Store, 16 million euros, now more than 10 pharma companies joining us, all the big ones, to make all data in the life sciences interoperable in this way. And we published an article in Nature Genetics about that in April last year, which was very well received, and we started this nanopub thing for people to play around. And we have already hackathons now for people from entirely different fields, as you see in the photograph on the right hand side, to use this. And we just published the nano-publication guidelines that everyone can download in a week from now, and you can decide whether you want to work with this or not with your own data set. So let me explain to you a little bit what this is. So this was the paper that we published, and there was an editorial in the same Nature Genetics by Miles Exton, the editor, saying we can now crowdsource human mutations. And this was about an article in the same Nature Genetics journal that was about single polymorphism databases. And we show that this can now promote crowdsourcing, because if people say this SNP in this gene is associated with this disease, they get credit for being a curator of that nano-publication. So a nano-publication is essentially a single meaningful assertion, its author, its time, date, stamp, very much, it's the smallest publication you can ever imagine. But now it is in RDF, so it is computer reasonable and computer readable. So that doesn't mean that we want to do away with the scholarly literature. Here we say, although we argue, this is part of the paper, that the narrative form of scholarly communication will continue to be needed in the future, we also recognize that data-intensive sciences need computer-readable information. And it follows that de novo claims and the supporting data should be exchanged in machine-readable, unambiguous form. But still we want to link out to the supporting papers, the databases and the data sets on which these triples that form ridiculograms are based. So this is a nano-publication as it is now in our new guidelines. Again, I won't show you any RDF, but I hope people have heard of RDF, the source description framework. You have a subject, S, predicate, object combination. And all three, in our minds, are concepts. So the classical semantic web usually doesn't treat the predicate as a concept, it's just a word, same as, or whatever. But we want to unambiguously identify, let's say this says malaria is transmitted by mosquitoes in the assertion part, then malaria transmission and mosquitoes are defined in UMLS, and if you find Plasmodium falciparum is transmitted by Anopheles gambiae, which is the mosquito that does it in Africa, you probably can associate these things through the ontology, but essentially an assertion is 3 UUIDs, totally non-semantic, so there is no semantics in the UUID, it's just a long number, so there is no temptation to ever change it, because the big thing about URIs is they're not stable, they change all the time. Even the DOI of articles, Crossref is struggling because British Medical Journal decides to change its name, they want to change the URI, and now all the web services using that URI fall down if you don't take measures. So the first part is the actual core assertion. This could be more than one triple. You say this SNP in this gene causes this disease, it's already more than one triple. So the smallest possible assertion could be multiple triples already, a triple being one SPO combination. That is wrapped in a small named graph in RDF, and then, very importantly, we treat it as a publication, so every triple that Semantic Medline would have would be one nano-publication. And now it has supporting evidence, like this statement is only true when the mosquito is female and you're in Africa because otherwise it's too cold, you know. So even a very truism, like malaria is transmitted by mosquitoes, according to the Barrett Smiths of this world, is not always true. And if one substance inhibits another substance, that's probably only true when the temperature is higher than a certain amount. So that's the supporting evidence. That's again triple. pH higher than literal 8 value, whatever, all based on ontologies. The provenance is extremely important, the green one, which is the authors. So every author, you're working closely with Vivo and ORCID to get the Open Researcher Contributor ID. We have already 1.7 million UUIDs of people in the concept wiki, and we refer to authors by their UUID as well. And we have an ARTA table, also referred to as Scopus ID, ISI, Social Security number, whatever you want to put there, but all referring to the same concept, so people are concepts as well. Obviously, since this is now a little named graph with UUIDs as nodes, you can hash an automatic nanopublication identifier, which is unique again, it's a UUID again. So now every nanopublication has again a unique UUID, so it's universally findable by a computer. And it has an integrity key, so you know that nobody tampered with your nanopublication. Okay? So this is a wrapped little container of triples, called a named graph in RDF, which is essentially, for lay people, just the smallest publication that makes sense that you can ever imagine. That is what a nanopublication actually is. It is completely semantic web compatible, but it is not comparable to what we currently see in the linked open data client. So we basically introduced the issue of what we call data publication. So you have a relational database, you have a data set, you have literature, you can republish that in nanopublication. Let me tell you a little bit about that. If you, for example, say cognitive deficits, UMLS concept, and base 1, a gene, since they relate to each other. That's the simplest predicate you could imagine, but even relate to, you could define as a UUID and say this is what we mean by this predicate. Then we have the provenance and the context. That is essentially one nanopublication. Obviously, these things get repeated on the web. If you look in Medline, and obviously Thomas will update that now, but when I last checked years ago, the statement malaria is transmitted by mosquitoes, we found plus 6,000 plus times in Medline abstracts alone. And the only thing that is different between all these identical assertions is the PubMed ID and the authors that repeated that statement as a citation or whatever. Now, obviously, the 6,000 first statement, malaria is transmitted by mosquitoes, you never read that. If I read an article about malaria, of course you skip all that. The computer doesn't need all this repetition, which makes things human readable. So if you would be able to now take all the redundancy out and say, okay, we found 6,000 times malaria is transmitted by mosquitoes in PubMed, we collapse that into what we call a cardinal assertion. That is the basic assertion malaria is transmitted by mosquitoes or cognitive deficits relate to this gene. We found 6,000 instances. Now we can, based on the provenance and the context, is this curated by a Swiss prot or not? Is it only a Medline? Is it only a prediction? We can get a summarizing, and the E stands for the evidence factor. So now we can relate one value, the evidence factor, to each statement. And malaria is transmitted by mosquitoes, we probably have 0.9999 because it's almost sure that this will never change. Guy got the Nobel Prize 100 years ago, probably it's true. Nothing is really true in science, of course, until the opposite is proven. If we have one of those predictions, 44,000 protein-protein interaction predictions that we have published, it will have a very low evidence factor because it's only once and it's only a computer prediction. So now, modern reasoners, next year, Lark will realize the first descriptive logic reasoner that is able to go over 200 billion of those triples every day, taking into account the weight of each of those connections. So a computer prediction will get a much lighter weight in the reasoning than something like malaria is transmitted by mosquitoes or a protein-protein interaction that is curated by Amos Byrock himself. But being able to check back is very important because if this cardinal assertion shows up in my radiculogram and it connects two very important clusters of concepts, I want to click on it, see the PubMed articles, read why I would believe this one statement, because apparently if I cut it, my two concept clouds will move apart and maybe my hypothesis is useless. So for mind-machine interaction, We need both human readability of individual nanopublications. We need the narrative literature to go back, and we argue all about that in the Nature paper, so I won't bother you very much with that now. And I know we are very much in agreement on this part, that we need the narrative text anyway as a background. So the estimate that we made when we started the Console Web Alliance, and this was made by Frank van Harmelen, the director of the Large Knowledge Collider, he said, we estimated if we have mined Medline, now we just heard how many triples do you have for Medline?
56 million.
56 million, that's Medline. But if we mine Swissprot, Campbell, all the data sets, and all microarrays that we know, we estimate that right now there is, but are these unique triples?
No, those are triples.
Yes. So basically, if you say we mine everything, we estimated that we will have 10 to the 14th nanopublications. Now that you cannot store that anywhere logically in one server, and certainly even Lark, the biggest, fastest reasoner, cannot handle that. However, if we simply estimate that the average statement, like malaria is transmitted by mosquitoes, you find hundreds of thousands of times on the web, is there 1,000 times on average, then the estimate is that we'll end up with the number of triples that is in the 10 to the 11th range, 100, 200 billion. And today, in the Netherlands alone, LARC, the Large Knowledge Collider, can do DL, descriptive logic, over 100 billion triples. So next year we will be able to do 200 billion. So essentially, if there is every 40 seconds a new PubMed article coming, and we mine that, and get databases and huge data sets, we easily have thousands of new nanopublications every day. And there is a new PubMed article every 40 seconds. And it contains a lot of redundancy, but there is also some new statements there. So these would come into the system, and it would spark LARC every day to reason over the entire knowledge in life sciences and deliver the new insights on a silver platter. That's the What we also did in the guidelines, you can read those guidelines yourself, is, we don't want to capture every single microarray data set in Geo, for example. So what you say, if you have raw data, and for example a 200 by 200 matrix of genes being overexpressed in any experiment, and for example the provenance is we compared melanoma cells with healthy cells, you don't want to create 200 by 200 nanopublications all with exactly the same provenance, saying this gene overexpressed in this context with this gene. So what we recommend is you just publish a data set, you just have individual associations in the data set, you wrap the whole thing as a data set, and you say the provenance and the context is for the whole set. If anyone uses any of the associations in that GWAS data or microarray data and turns it to another publication, you just attach that provenance to it and it has this problem. So this is something we call pre-nano publication of big data sets. Well, if you have time later, Albert can show that we are now already building some interfaces where you can go to a Medline abstract, click a few concepts you're interested in, get the people that know most about it, they position themselves in the space close to these concepts. We know all the synonyms from UMLS, and if you click on one of these people, you can just call them by Skype if they're green for you. And we have that for 1.7 million people already in the life sciences. Of course, right now, for most of them, we guessed their Skype ID, if they have one, but I took one of my guys that moved to New York, and the system picked perfectly his new Skype ID. However, sometimes you see 3 or 4 of them, and you can have a click box and say, yeah, that's me. So now you are part of this consulting intellectual networking system to deal with these elements. So that is the technology. Let me finalize with a few slides on how this fits. Here you see that we have, in the Netherlands alone, we have all these centers: Metabolomic Center, Proteomic Center, Center for Genome Diagnostics, MBIC, where I work, Systems Biology. We have started this thing DISC now because we said we have to do this together. And in Europe, you have Elixir now, Data for Life, which is the bioinformatics research infrastructure in Europe. I'm in the board of that, and it supports all kinds of huge European projects like Eurobio imaging, Beatrice for clinical trials, ISBE for systems biology, BBMRI is the biobanking, ECRIN is clinical trials, and Instruct is for structural proteins. All these will spit out terabytes, petabytes, yottabytes of data in the next few years. And unless we learn how to deal with it, we will just come to a grinding halt. I'm going to China in 2 weeks from now. They are going to China to sequence very soon 1 million Chinese people. They want to connect their sequence to their Chinese medical record and talk to the UMLS content from a Chinese token for a disease. It should go to the UUID that knows which UMLS concept that is.
If there are any chemists in the room, a yottabyte is 2 times Avogadro's number. Usually viewed as the largest number that has scientific meaning, and now here they are talking about it.
Exactly. So obviously we do this not alone, so as you can see here, there's lots of people involved in my Dutch team, but also this concept of Alliance and Open Pharmacological Space project. We are very grateful that this is a sort of the launching project of the Nano Publication ID, with all these people from different companies being involved, and it's growing very fast. Merck, we are going to Boston tonight to talk to Merck again. They want to join Merck America then. Johnson & Johnson just signed up, and so on. So this is really going to be a universal standard for the pharma. But ending, and then switching to the other talk, is this is obviously essentially the way we think. We associate concepts, and if you remember this chaos, edge of chaos, Sawtooth, in fact knowledge discovery is discovering more and more patterns in what we originally thought was chaos. And the more knowledge we have, the more patterns we see, the more associations we make and turn them into knowledge. And that is universal for every science, not just life sciences. But I would imagine that anyone who wants to start with this, if there is any urgency, it's in the life sciences, and then we should all team together to get this done. And that is essentially what I wanted to pitch here.
I'd like your final comment, Baron, that this is a prototype for all science and for areas beyond science, and bioscience is a clear leader in bringing in this dimension of the future.
Because it's really the urgency and the reason the pharma put collective money into this. The IMI initiative, Innovative Medicines Initiative, is €1 billion from the European Commission, $1 billion from the pharma. The reason, as we discussed before lunch, is that they used to do this all in the house behind their firewall 22 times over. They simply don't have the money anymore, because, as you know, they're all going basically down the drain. And so they at least said, let's do this together, give it to a public-private partnership, and we won the call for that. And we are now creating this open pharmacological space. First internal release after a year will be in March this year. And in September it will go online for everyone to use, download. It's completely open source, open access. And I wouldn't have believed that 2 years ago that the pharma would ever do that. But they are forced to do it. And the good news is that we have big white and green technology in the Netherlands, like the Monsanto's and Rijkswaan, you know, the green genetics guys, and DSM and all the big biotechnology companies. They have still enormous amounts of money. But they have told me, oh, we need this as well for biotechnology, we need it for green genetics. We have 5,000 tomato genomes in the Netherlands because we wanted to keep ahead and produce tomatoes to export them to Mexico. And so, so it's true for all the other sciences as well. So the pharma, pure out of necessity, not being able to find new drugs anymore, have now started this. But they also tell me, all these guys you see here, like Ian Dix and so on from AstraZeneca, what we are so scared about is that your system will now reveal complexity to the level that we will see. We won't find any new drugs based on single small molecules. Just a final scary thing, they just disturbed yeast cells, of which we know virtually all pathways in the cell. They disturbed them repeatedly until they almost died, so they put a lot of selective pressure. Then they grew up the surviving cells, and they showed, Larry Hunter told us, that 40% of all pathways have changed fundamentally. So we know that in every molecular pathway there is at least 2 or 3 redundancies in the proteins that if you kill one of them, they just switch to another. That's why 90% of the small molecules don't work. But we now see the mechanism. So now at least we know why we don't find any drugs. That doesn't mean we have the solution to find new drugs. So if we find new drugs, they will be multi-component products, lots of IP associated. So valorizing or commercializing these things will be a nightmare. Right.
Wonderful.
Anyway.
Thank you very much. I call for a 2-minute break for those of us who drank too much ice cream.
Meanwhile, any questions? I'm here. I didn't bring any.
So,
Everything's way.
would you, and this is a very exciting, work, of course, and very necessary. Right? There is a huge component that's will link the phenotypes to the pathways, and that will probably increase the magnitude of this with the new behavioral measurements that are coming up, or which bio people ignore.
Well, the thing is, the phenotypes, if we stick to human phenotypes, then of course ICD-11 is now under development and will be part of this whole thing. But if you go across organism, and you look at the phenotypes from zebrafish to man, it's an enormous complexity.
Yeah, but even in human phenotype, because the issues that we face dealing with diseases like neurological diseases, right? Then they're very complex. They're much more complex than you least.
Yeah.
And even that, right? And the data that we are now beginning to acquire, behavior data, what people know, what people do, and which are very important for neurological, going to be even more, make it more complicated, in particular because the provenance of the data and the description of the data is so much more difficult than in the past ways.
But you need to really understand that we do this already for diseases and so on. And even way outside the life sciences in the humanities. So you can even look at semantic drift of a concept over time.
Yes.
You know, what did the term mean 100 years ago, and what's the constant cloud around it? So the real connection between a molecular, I don't see a fundamental difference between a molecular pathway and the description of a complex neurological disease and how it relates to schizophrenia and probably this particular behavior. That is something you can wrap in an RDF graph, and then you have narrative text connected to that to read the background data and stuff, and that's what we both need.
Right.
So this is not a universal solution for everything.
No,
We still
no,
need the narrative.